Hello everyone, welcome back to the class of biotechnology. Today we are going to discuss a very important enzyme, that is alkaline phosphatase. In short, you can call it as ALP. This enzyme has a various different names, that is the alkaline phosphohydrolase, alkaline phenyl phosphatase, orthophosphoric monoester phosphohydrolase. Alkaline phosphatase are a group of isoenzymes. What is isoenzymes? Isoenzymes are those enzymes that are having a different forms of same enzymes. Means these enzymes are having different forms, but all the forms catalyzes the same chemical reaction, right? Means they catalyzes the same chemical reaction but exhibit different physical or biochemical properties. It is also a class of homodimeric protein enzyme. Homodimeric means ALP backbone is composed of two identical protein subunits. Means suppose this is one subunit and this is another subunit, and these two subunits are identical to each other. These two monomeric unit, or you can say subunit, held together by a covalent bond, and this covalent bond might be hydrogen bond, ionic bond, hydrophobic bond, van der Waals force. The weight of the ALP is approximately around 85 kilodalton, and ALP are the plasma membrane bound glycoprotein. Means it carry a side chain of carbohydrates. ALP is found in cytosol of liver cells and membrane of hepatocytes, and in some less concentration it is also present in placenta, mucosa, kidney and bone. So these are the location where you can find the ALP enzyme. Zinc, magnesium are the essential cofactor for the activity of the ALP. So this was all about the basic background or the basic introduction of the ALP, that what it is and where it is found. But if we talk about its application, then ALP is a versatile enzyme. That means it is having a wide range of application in various fields, including the clinical diagnostic research and industry. ALP also play important role in recombinant DNA technology. In this lecture, we are going to discuss specifically about the role of the ALP in the field of recombinant DNA technology, that is RDT. So in the field of recombinant DNA technology, its physiological role is in the removal of the phosphate by the action of the hydrolysis from the ends of the nucleic acid. Actually, it removes the phosphate from the 5' end. Here is phosphate present at the 5' ends. So it can remove the phosphate from the 5' ends of the any molecule. Specifically in the recombinant technology, if we talk, we deal with the double-stranded DNA, right? We talk about the double-stranded DNA. By the action of the ALP, we can easily remove the phosphate from the 5' end. And why we want to remove the phosphate from the 5' end? Actually, it prevents the re-circularization during the cloning by removing the single phosphate group from the 5' end of the linear vector. Another important factor that should be considered is the alkaline pH. That means at the alkaline pH, or for the removal of the phosphate group from the 5' ends of the double-stranded DNA, the condition of the environment should be alkaline. If the pH of the solution or the buffer in which double-stranded DNA is suspended, that pH should be alkaline in nature. Only in alkaline condition, ALP can efficiently remove the phosphate from the 5' end of the double-stranded DNA. And you will get a double-stranded DNA that lacks the phosphate group from the 5' ends and leaving behind the only free 3' hydroxyl group. There is no any free phosphate group at the 5' ends, right? So this DNA lacks the phosphate group. It lacks the free, actually it is a free phosphate. So it lacks the free phosphate group at the free 5' ends. and it contains only the free hydroxyl group at both the strands of the double stranded DNA, right? So now let us understand this whole concept, that how it works and what is the application and where it is being important by using this particular diagram. Suppose this is a bacteria and this bacteria contain a plasmid DNA and this is a genomic DNA. Now when you isolate this plasmid from the bacteria, and you all know that this is a circular plasmid, right? And this plasmid must be having a some restriction site, and when you digest this particular plasmid by using a some restriction enzyme that is specific to this restriction site, then you will get the plasmid like this. This is a digested plasmid, okay? And now you want to incorporate, or you can say you want to combine this bacterial plasmid with some any other gene. Suppose this is a gene of a human and you want to incorporate this particular G2 gene. Suppose this is gene, this particular gene you want to incorporate into this particular bacterial plasmid, and for that you use an enzyme named DNA ligase. We have already discussed about the DNA ligase, that how it actually joins the two different fragments together. So this is a bacterial plasmid and this is a some any other gene of any other organism, and you want to join these both together. So to join these two segments of DNA, that is one from the bacteria and another from the human, we put these DNA into a test tube containing a buffer, so to maintain the alkaline pH, and then into that we added the DNA ligase into it. DNA ligase, you already know that it requires free OH and free phosphate, right? It requires free OH and free phosphate group. And by using this free phosphate and free OH, it forms the phosphodiester bond. We have already discussed the mechanism of action of the DNA ligase in our last lecture. So by using these two groups, that is the phosphate and OH, it forms the phosphodiester bond, and in this way it sealed this particular region, right? So here you are having a free OH and free phosphate group on both the ends of the DNA. Here also you are having free phosphate, free OH and free OH, and in this particular part of gene of interest which you want to ligate into a bacterial vector, this particular gene of interest is also having a free OH and free phosphate group at its 5' and 3' ends. So due to the presence of free OH and free phosphate group on both the DNAs, there arises the two possibilities. One is that the bacterial DNA, due to having the free 5' phosphate group and 3' OH group, religate or rejoins its ends like this. It may rejoin together without containing or without attaining this particular gene of interest into it. Such type of plasmid is called the self-ligated plasmid, and this is the condition first of the two possibilities. The next possibility is that it contains or it carry on or it incorporates the gene of interest which you want to incorporate into it. It incorporated that particular gene of interest into it, because this gene of interest is also having free OH and free phosphate group, right? So this OH group will form a phosphodiester bond with this particular phosphate group, and this particular phosphate group will form a phosphodiester bond with this particular hydroxyl group. So in this way there is a formation of the two phosphodiester bonds at both the ends, right? And in this manner this particular segments of the gene of interest ligated into the bacterial vector. Such type of plasmid, after the addition of the gene of interest or such type of vector that carries the gene of interest into it are called the recombinant DNA. Our motive is to obtain this particular recombinant DNA only because it carries the gene of interest into it. And we want to avoid this situation because it is not carrying the gene of interest, right? So to avoid this condition or to avoid this particular possibility, here comes the role of ALP. So ALP under alkaline condition removes the phosphate group from the 5' end, right? ALP removes this particular phosphate group from the 5' end. So in the presence of the ALP, these two phosphate will be removed from the 5' end. So before adding to the gene of interest to the plasmid, before adding this particular gene of interest to the plasmid vector of bacteria, we first treat this vector, this bacterial vector with ALP under alkaline condition. And ALP here removes the phosphate group from the 5' end and leaving behind the vectors lacking the phosphate group. Here we are not having any phosphate group, right? So in this particular vector, there is only a hydroxyl group present at the 3' end, but there is a no phosphate group at the 5' end. Hence, in this way, because of the absence of the phosphate group, there is a no phosphodiester bond formation, right? Because of the absence of the phosphate group, there is no phosphodiester bond formation because phosphodiester bond is formed only in between the free phosphate and free hydroxyl group, right? So because of the absence of this particular phosphate group at the 5' end, there is a no phosphate bond formation, right? So due to this, you can avoid the chance of self ligation or rejoining, right? But when we see here in this gene of interest from the human source, there is a phosphate group at the 5' end, right? Because here we did not treat this particular gene of interest with ALP. We have only treated this plasmid vector with ALP, right? So there is a presence of phosphate group at the 5' end of the particular gene of interest from the human source. So this particular phosphate group will definitely make a phosphodiester bond when it comes in contact proximity with the OH. So this phosphate group will definitely form a phosphodiester bond with this hydroxyl group, and this particular phosphate group will form a phosphodiester bond with this particular OH, that is hydroxyl group, and hence in this way there is a formation of the phosphodiester bond here, right? So the addition of this gene of interest with this bacterial plasmid vector in the presence of the DNA ligase resultant into the ligated plasmid vector that carries the gene of interest, and this type of ligated plasmid vector that carries the gene of interest is called the recombinant DNA, right? This is the gene of interest. In this way, you can only add or ligate your particular gene of interest into it by avoiding the self ligation at the same time. So this was the role of ALP, by which you can avoid the chance of self ligation and you can increase efficiency of the formation of the recombinant DNA. So this was all about the alkaline phosphatase. Thank you so much. If you like this video, please like and subscribe, and please don't forget to hit the notification bell so that you cannot miss any of the future video. In the next lecture, we are going to discuss about the basic behind the clonings, that what are the basic steps that we should know and what are the basics actually we should be aware of before proceeding to the steps of cloning. So thank you so much.